Hello and welcome dear students. Today we are going to discuss about the topic translation or protein synthesis. The main objectives of today's lecture are to study the basic idea of translation, to study about various enzymes involved in protein synthesis, to learn the mechanism of protein synthesis, to know the critical steps involved in the prokaryotic and eukaryotic translation mechanism. Dear students, let's first start with the introduction to translation. The pathway of protein synthesis is called translation because the language of the nucleotide sequence on the mRNA is translated into the language of the amino acid sequence. The mRNA is translated from its 5' end to its 3' end, producing a protein synthesized from its amino terminal end to its carboxylic terminal end. So the question, what is protein synthesis? Protein synthesis is a process of synthesizing proteins in a chain of amino acids known as polypeptides. It is the second part of the central dogma in genetics after the transcription. It takes place in the ribosomes found in the cytosol or cytoplasm or those attached to the rough endoplasmic reticulum. The functions of the ribosome are to read the sequence of the codons in mRNA and the tRNA molecules that transfer or transport or bring the amino acids to the ribosomes in the correct sequence. However, other molecules are also involved in the process of translation, such as various enzymatic factors. The translation process involves reading the genetic code in mRNA to make proteins. The entire translation process can be summarized into 3 phases, that is the initiation, elongation and termination. Dear students, let's discuss about the protein synthesis machinery in the cell. The translation process is aided by 2 major factors. A translator, this is the molecule that conducts the translation. Substrate, this is where the mRNA is translated into a new protein. That is the translator desk. The translation process is guided by machinery composed of ribosomes. Ribosomes are made of ribosomal rRNA and proteins, and therefore they are also named as ribozymes because the rRNA has enzymatic activity. The rRNA has the peptidyl transferase activity that bonds the amino acids. The ribosomes have 2 subunits of rRNA and proteins, a large subunit with 3 active sites, that's EPA, which are critical for the catalytic activity of ribosomes. The other one is the transfer RNA or tRNA. Each tRNA has an anticodon for the amino acid codon it carries, which are complementary to each other. For example, lysine is coded by AAG, and therefore the anticodon that will be carried by tRNA will be UUC. Therefore, when the codon AAG appears, an anticodon UUC of tRNA will bind to it temporarily. When tRNA is bound to mRNA, the tRNA then releases its amino acid. rRNA then helps to form bonds between amino acids as they are transported to the ribosomes one by one, thus creating a polypeptide chain. The polypeptide chain keeps growing until it reaches a stop codon. Dear students, let's now discuss about the protein synthesis enzymes and functions. Peptidyl transferase is the main enzyme used in translation. It's found in the ribosomes with an enzymatic activity that catalyzes the formation of a covalent peptide bond between the adjacent amino acids. The enzyme's activity is to form a peptide bond between adjacent amino acids using tRNA during translation. The enzyme's activity uses two substrates, of which one has the growing peptide chain and the other bears the amino acid that is added to the chain. It is located in the large subunit of the ribosomes, and therefore the primary function of peptidyl transferase is to catalyze the addition of amino acid residues, allowing the polypeptide chain to grow. The peptidyl transferase enzyme is entirely made up of RNA, and its mechanism is mediated by ribosomal RNA, which is a ribozyme made up of ribonucleotides. In prokaryotes, the 23S subunit contains the peptidyl transferase between the A site and O site of tRNA, while in eukaryotes it is found in the 28S subunit. Dear students, let's now have an overview of the protein synthesis. The ribosomal translation is initiated when the ribosomes recognize the starting point of mRNA, where it binds a molecule of tRNA that bears a single amino acid. In prokaryotes, the initial amino acid is N-formyl methionine. During elongation, the second amino acid is linked to the first one. The ribosome then shifts its position on the mRNA and repeats the elongation cycle. When the elongation process reaches the stop codon, the amino acid chain folds spontaneously to form a protein. The ribosomes then split into two subunits but later rejoin before another mRNA is translated. Protein synthesis is facilitated by several catalytic proteins, which include initiation, elongation, termination factors, and guanosyl triphosphates, that is GTPs. GTP is a molecule that releases energy when converted into guanosine diphosphate or GDP from GTP. Dear students, let's discuss the prokaryotic translation process. Translation involves translating the sequences of the mRNA, that is messenger RNA molecule, to a sequence of amino acids during protein synthesis. It is the process in which ribosomes in the cytoplasm or endoplasmic reticulum synthesize proteins after the process of transcription of DNA to RNA. The ribosomes exist normally as a separate subunit that are composed of protein and rRNA. The subunits come together to form a ribosome when they bind to an mRNA near its 5' end. On binding to an mRNA, the ribosome reads the nucleotide sequence from the 5' to 3' direction, synthesizing the corresponding protein from amino acids in an N-terminal, amino terminal to C-terminal, that is carboxyl terminal direction. Ribosomes are located in the cytosol, either freely floating or associated with the endoplasmic reticulum. They serve to synthesize proteins. Dear students, let's now understand the ribosomal sites of protein translation. Each prokaryotic ribosome, shown schematically, has 3 binding sites for tRNA. The amino acyl tRNA binding site, or A site, is where during elongation the incoming amino acyl tRNA binds. The peptidyl tRNA binding site, or P site, is where the tRNA linked to the growing polypeptide chain is bound. The exit site, or E site, is a binding site for tRNA following its role in translation and prior to its release from the ribosome. All 3 sites, A, P, and E, are formed by the rRNA molecules in the ribosome. Dear students, let's now discuss the process of translation. Protein synthesis or translation takes place in 3 stages, that's initiation, elongation, and termination. During initiation, the mRNA ribosome complex is formed and the first codon, always AUG, binds the first amino acyl tRNA called initiator tRNA. During the elongation phase, the other codons are read sequentially and the polypeptide grows by addition of amino acids to its C-terminal end. This process continues until a termination codon or a stop codon which does not have a corresponding amino acyl tRNA with which to base pair is reached. At this point, protein synthesis ceases, that is the termination phase, and the finished polypeptide is released from the ribosome. Dear students, let's now discuss the synthesis of amino acyl tRNA. Synthesis of amino acyl tRNAs is crucially important for two reasons. Each amino acid must be covalently linked to a tRNA molecule in order to take part in protein synthesis, which depends upon the adapter function of tRNA to ensure that the correct amino acids are incorporated. The covalent bond that is formed between the amino acid and the tRNA is a high energy bond that enables the amino acid to react with the end of the growing polypeptide chain to form a new peptide bond. For this reason, the synthesis of amino acyl tRNA is also referred to as amino acid activation. Each tRNA molecule has a clover leaf secondary structure with the anticodon accessible at the end of the anticodon stem loop. During synthesis of the amino acyl tRNA, the amino acid is covalently bound to the A residue of the CCA sequence at the 3' end. Each tRNA molecule carries only a single amino acid. The attachment of an amino acid to a tRNA is catalyzed by an enzyme called amino acyl tRNA synthetase. A separate amino acyl tRNA synthase exists for every amino acid, making 20 synthetases in total. The synthesis reaction occurs in two steps. The first step is the reaction of an amino acid and ATP to form an amino acyl adenylate, also known as amino acyl AMP. In the second step, without leaving the enzyme, the amino acyl group of amino acyl AMP is transferred to the 3' end of the tRNA molecule to form amino acyl tRNA. The overall reaction is amino acid plus ATP plus tRNA. It gives to amino acyl tRNA plus AMP plus 1 phosphate. Dear students, let's now discuss the initiation of protein synthesis. The first codon translated in all mRNAs is the start codon or initiation codon AUG, which codes for methionine. Two different tRNAs are used for the two types of AUG codon. One is the tRNA formylmethionine is used for the initiation codon and is called the initiator tRNA, whereas tRNA M-methionine is used for internal AUG codons. In prokaryotes, the first amino acid of a new protein is N-formylmethionine, abbreviated FMET. Hence, the amino acyl tRNA used in the initiation is FMET, tRNA FMET. A short sequence rich in purines at the 5' to 3' end like AGG, AGGU called the Shine-Dalgarno sequence lies 5' to the AUG initiation codon and is complementary to part of the 16S rRNA in the small ribosomal subunit. Therefore, this is the binding site for the 30S ribosomal subunit, which when migrates in a 3' direction along the mRNA until it encounters the AUG initiation codon. Initiation of protein synthesis requires proteins called initiation factors. In prokaryotes, 3 initiation factors like IF1, initiation factor 2, and IF3, that is initiation factor 3, are essential. Because of the complexity of the process, the exact order of binding of initiation factor 1, initiation factor 2, initiation factor 3, formylmethionine, tRNA formylmethionine is controversial. The steps involved in the protein synthesis are the initiation, elongation, and termination. The initiation begins with the binding of IF1 and IF3 to the small subunit of ribosome, that is 30S. Their role is to stop the 30S subunit binding to the 50S subunit in the absence of mRNA and formylmethionine tRNA formylmethionine, which would result in a non-functional ribosome. The small subunit then binds to the mRNA via the Shine-Dalgarno sequence and moves 3' along the mRNA until it locates the AUG initiation codon. The initiator tRNA charged with N-formylmethionine and in a complex with initiation factor 2 and GTP now binds. Initiation factor 3 is released. The complex of mRNA, formylmethionine, tRNA, formylmethionine, initiation factor 1, initiation factor 2, and the 30S ribosomal subunit is called the 30S initiation complex. The large 50S ribosomal subunit now binds with the release of initiation factor 1, initiation factor 2, and hydrolysis of GTP to form a 70S initiation complex. Dear students, the second step is the elongation in protein synthesis. At the start of the first round of elongation, the initiation codon, that is AUG, is positioned in the P site with formylmethionine tRNA formylmethionine bound to it via codon anticodon base pairing. The next codon in the mRNA is positioned in the A site. Elongation of the polypeptide chain occurs in 3 steps called the elongation cycle, namely amino acyl tRNA binding, peptide bond formation, and translocation. Let's start the first one, amino acyl tRNA binding. The corresponding amino acyl tRNA for the second codon binds to the A site via codon anticodon interaction. Binding of the amino acyl tRNA requires elongation factor EFTU and GTP, which bind as an amino acyl tRNA EFTU GTP complex. Following binding, the GTP is hydrolyzed and the elongation factor TU is released, now bound to the GDP. Before the elongation factor TU molecule can catalyze the binding of another charged tRNA to the ribosome, it must be regenerated by a process involving another elongation factor, that's elongation factor TS. This regeneration is called the EFTU-EF-TS exchange cycle. First, elongation factor TS binds to EFTU and displaces the GDP. Then GTP binds to the elongation factor TU and displaces EFTS. The EFTU GTP is now ready to take part in another round of the elongation cycle. The second step in the elongation is the formation of a peptide bond. So the second step of peptide bond formation is catalyzed by peptidyl transferase enzyme. In this reaction, the carboxyl end of the amino acid bound to tRNA in the P site is uncoupled from the tRNA and becomes joined by a peptide bond to the amino group of the amino acid linked to the tRNA in the A site. The third step is the translocation in elongation. In the third step, a complex of elongation factor EFG, also called translocase, and GTP, that is EFG/GTP, binds to the ribosome. 3 concerted movements now occur, collectively called translocation. The deacylated tRNA moves from the P site to the E site, the dipeptidyl tRNA in the A site moves to the P site, and the ribosome moves along the mRNA from 5' to 3' by 3 nucleotides to place the next codon in the A site. During the translocation events, GTP is hydrolyzed to GDP and inorganic phosphate, and elongation factor G is released, ready to bind more GTP for another round of elongation. After translocation, the A site is empty and ready to receive the next amino acyl tRNA. The A site and the E site cannot be occupied simultaneously. Thus, the deacylated tRNA is released from the E site before the next amino acyl tRNA binds to the A site to start a new round of elongation. Elongation continues, adding one amino acid to the C-terminal end of the growing polypeptide for each codon that is read with the peptidyl tRNA, moving back and forth from the P site to the A site as it grows. Dear students, let's now discuss about the last step of protein synthesis, that is the termination. Eventually, one of three termination codons, also called stop codons, becomes positioned in the A site. These are UAA, UAA and UGA, also called Embr, Ocher and Opal. Unlike other codons, prokaryotic cells do not contain amino acyl tRNAs complementary to stop codons. Instead, one of the two release factors, that is RF1 and RF2, binds instead. RF1 recognizes UAA and UAG, whereas RF2 recognizes UAA and UGA. A third release factor, RF3, is also needed to assist RF1 and RF2 interaction with the ribosome. Thus, either RF1 plus RF3 or RF2 plus RF3 bind depending on the exact termination codon in the A site of the tRNA. RF1 or RF2 bind at or near the A site, whereas RF3 GTP binds elsewhere on the ribosome. The release factors cause the peptidyl transferase activity to transfer the polypeptide to a water molecule instead of amino acyl tRNA, effectively cleaving the bond between the polypeptide and tRNA in the P site. The free polypeptide now leaves the ribosome, followed by the mRNA and free tRNA, and the ribosome dissociates into 30S and 50S subunits, ready to start translation again. Dear students, let's now discuss the protein synthesis in eukaryotes. The eukaryotic translation includes first the ribosomes. The ribosomes exist normally as separate subunits that are composed of protein and rRNA. Eukaryotic ribosomes are larger, that is the 80S, and more complex than prokaryotic ribosomes, that is 70S. The subunits come together to form a ribosome when they bind to an mRNA near its 5' end. On binding to an mRNA, the ribosome reads the nucleotide sequence from the 5 to 3 direction, synthesizing the corresponding protein from amino acids in an N-terminal, that is amino terminal, to C-terminal, carboxyl terminal direction. Ribosomes are located in the cytosol, either freely floating or associated with the endoplasmic reticulum. They serve to synthesize proteins. Protein synthesis process. The overall mechanism of protein synthesis in eukaryotes is basically the same as in prokaryotes. However, there are some significant differences. Whereas a prokaryotic ribosome has a sedimentation coefficient of 70S and subunits of 30S and 50S, an eukaryotic ribosome has a sedimentation coefficient of 80S with subunits as 40S and 60S. The composition of eukaryotic ribosomal subunit is also more complex than prokaryotic subunits, but the function of each subunit is essentially the same as in prokaryotes. In eukaryotes, each mRNA is monocistronic, that is, discounting any subsequent post-translational cleavage reactions that may occur. The mRNA encodes a single protein. In prokaryotes, many mRNAs are polycistronic, that is, they encode several proteins. Each coding sequence in a prokaryotic mRNA has its own initiation and termination codons. Initiation of protein synthesis in eukaryotes requires at least 9 distinct eukaryotic initiation factors, compared with the 3 initiation factors IFs in prokaryotes. In eukaryotes, the initiation amino acid is methionine, not formyl methionine as in prokaryotes. As in prokaryotes, a special initiation tRNA is required for initiation and is distinct from the tRNA that recognizes and binds to codons for methionine at internal position in the mRNA. When charged with methionine ready to begin initiation, this is known as methionine tRNA. The main difference between initiation of translation in prokaryotes and eukaryotes is that in bacteria, a Shine-Dalgarno sequence lies 5' to the AUG initiation codon and is the binding site for the 30S ribosomal subunit. In contrast, all eukaryotic mRNAs do not contain Shine-Dalgarno sequences. Instead, a 40S ribosomal subunit attaches at the 5' end of the mRNA and moves downstream, that is in 5' to 3' direction, until it finds the AUG initiation codons. This process is called scanning. Prokaryotic translation requires no helicases, presumably because protein synthesis in bacteria can start even as the mRNA is still being synthesized, whereas in eukaryotes, transcription in the nucleus and translation in the cytoplasm are separate events that allow time for mRNA's secondary structure to form. Protein synthesis takes place in 3 stages, as in prokaryotes. In eukaryotes also, like initiation, elongation and termination. Let us discuss the initiation of protein synthesis in eukaryotes. The first step is the formation of a pre-initiation complex consisting of the 40S small ribosomal subunit, that is methionine, tRNA, MET, elongation factor F2 and GTP. The pre-initiation complex binds to the 5' end of the eukaryotic mRNA, a step that requires elongation factor 4F, also called cap binding complex, and elongation factor F3. The elongation factor 4F complex consists of elongation factor 4A, elongation factor -4E, and elongation factor 4G. ELF 4E binds to the 5' cap on the mRNA, while as elongation factor 4G interacts with the poly A binding protein on the poly A tail. The elongation factor 4A is an ATP dependent RNA helicase that unwinds any secondary structures in the mRNA, preparing it for translation. The complex then moves along the mRNA in 5' to 3' direction until it locates the AUG initiation codon, that is the scanning process. The 5 untranslated regions of eukaryotic mRNA vary in length but can be several hundred nucleotides long and may contain secondary structures such as hairpin loops. These secondary structures are probably removed by initiation factors of the scanning complex. The initiation codon is usually recognizable because it is often but not always contained in a short sequence called the Kozak consensus, that is 5-ACCAUGG3-. Once the complex is positioned over the initiation codon, the 60S large ribosomal subunit binds to the form an ATS initiation complex, a step that requires the hydrolysis of GTP and leads to the release of several initiation factors. Dear students, let's discuss the elongation of protein synthesis in eukaryotes. Elongation depends on eukaryotic elongation factors. 3 elongation factors like EF1A, EF1B and EF2 are involved, which have similar functions to their prokaryotic counterparts: elongation factor TU, elongation factor TS and elongation factor G. At the end of the initiation step, the mRNA is positioned so that the next codon can be translated during the elongation stage of protein synthesis. The initiator tRNA occupies the P site in the ribosome and the A site is ready to receive an amino acyl tRNA. During chain elongation, Each additional amino acid is added to the nascent polypeptide chain in a 3-step microcycle. The steps in this microcycle are positioning the correct amino acyl tRNA in the A site of the ribosome, forming the peptide bond and shifting the mRNA by one codon relative to the ribosome. Although most codons encode the same amino acids in both prokaryotes and eukaryotes, the mRNA is synthesized within the organelles of some eukaryotes use a variant of the genetic code. During elongation in bacteria, the deacylated tRNA in the P site moves to the E site prior to leaving the ribosome. In contrast, although the situation is still not completely clear in eukaryotes, the deacylated tRNA appears to be ejected directly from the ribosome. Dear students, let's now talk about the termination of protein synthesis in the eukaryotes. Termination of elongation depends on eukaryotic release factors. In eukaryotes, eukaryotic release factor ERF1 recognizes all 3 termination codons, that's UAA, UAG and UGA, that's Ochre, Amber, Opal. And with the help of protein ERF3 terminates translation. Upon termination, the ribosome is disassembled and the completed polypeptide is released. Dear students, it was all about today's lecture about translation in prokaryotes and eukaryotes. Hope you have enjoyed it and understood it well. Take care. Goodbye.